Hello everybody, again my name is Logan Thomas and I'm here and happy to talk to you today about the relative risk that Aoudad might pose to bighorn sheep regarding pneumonia transmission. Pneumonia is a population-limiting disease in bighorn sheep for two primary reasons. First, when a naive population is exposed, we see a wide range of all-age mortalities, and it's even been recorded to wipe out some herds entirely. Second, when exposed adults do survive, the ewes in particular can be chronically infected with these pneumonic pathogens and serve to transmit them to their lambs for several years. This is why we see reduced lamb crops in both new and previously exposed herds for years to come. Pneumonia in bighorn sheep starts when the upper respiratory tract's normal pathogen clearing functions are inhibited. These changes can be started by risk factors like poor nutrition and environmental stressors. Alternatively, if the animals are exposed to new viruses or have viral flare-ups, this can certainly start causing these changes as well. We often see that stress and viral flare-ups go hand in hand, and they can certainly interact to start setting up this environment. However, the bacterial pathogen Mycoplasma ovonemoniae is particularly good at setting up this pneumonic environment and is considered a primary causative agent. The changes that we see in the upper respiratory tract allow bacteria normally found in the lungs to become pathogenic, and it also allows environmental pathogens that are typically cleared to hang around and cause damage. Pastorella species can act as either normal constituents or unwelcome pathogens for bighorn sheep, but environmental bacteria like Trupperella pyogenes and Fusobacterium necrophorum are good examples of what we see of the bacteria that are seen causing pneumonia through decreased pathogen clearing. The result of this is bronchopneumonia characterized by cranioventral suppurative inflammation and subsequent all-age die-offs immediately, and then our chronic effects of also having our subsequent poor lamb recruitment for years to come. So now that we know why pneumonia is an issue for bighorn sheep, let's address the reasons that we have to believe that Aoudad might serve as competent pathogen transmitters to bighorn sheep. If we look at the Capernet subfamily, we can see that Aoudad, Ammotragus lervia, are related to bighorn sheep through 6 evolutionary divergence events. I also want to draw attention to domestic goats, domestic sheep, and mountain goats. These 3 species are capable of harboring, shedding, and transmitting pathogens important in the etiology of bighorn sheep pneumonia, and interactions with these species are considered risks for pneumonia outbreaks in bighorn sheep. While Aoudad are more distantly related to bighorn sheep than are these other 3 species, their place within the Capernet subfamily still warrants investigation as to their pathogen competence. Aoudad were introduced to Texas from the Barbary Coast of Africa in the late 1950s. Most individuals were initially translocated to the Caprock Canyon area of Texas, but we can see here on the map that there are a few smaller translocation and dispersal events in the Trans-Pecos region of Texas, where they are now sympatrically occurring with desert bighorn sheep in Texas. Beyond their sympatric occurrence and evolutionary relationships to bighorn sheep, Aoudad also have several indications that their niches overlap with bighorn sheep. Specifically, we see that they both tend to occur on precipitous slopes at high elevations, share a notable portion of each other's diets, and are transiently seen occurring at the same artificial water guzzler sources. Conceptually, this provides ample opportunity for potential disease transmission events, either directly or indirectly, and this is depicted in the overlap that we see between the bighorn sheep and Aoudad niche curves. We believe that taken together, This knowledge warranted investigating whether Aoudad pose a pneumonia risk to bighorn sheep. Specifically, we wanted to determine whether Aoudad are susceptible to isolated mycoplasma compared to domestic sheep nasal washings that contained both mycoplasma and Pasteurella species that are normally found in the lungs of healthy domestic sheep. We wanted to see how Aoudad shed these pathogens in relation to their antibody responses, clinical health indications, and time since exposure. We also wanted to determine whether the presence of Pasteurella species influenced how mycoplasma was shed by Aoudad. To address these objectives, we caught 21 wild Aoudad via helicopter gun and transported them to Texas A&M's Veterinary Medical Research Park. 6 of these animals were assigned to the MOVI group that were inoculated with only isolated Mycoplasma pneumoniae. 5 animals in the wash group received nasal washes containing potentially important Pasteurella species, as well as Mycoplasma pneumoniae, and these were nasal washings from domestic sheep. 3 animals were assigned to the control group that were not inoculated with anything, but were allowed fence line contact to both of our treatment groups. Each day, animals were assigned clinical health scores based on aspects on coughing, nasal discharge, weakness, ear paresis, diarrhea, and head shaking. Sampling occurred approximately weekly for each group, for 5 and 6 weeks respectively for the wash group and the MOVI group. At each sampling event, we collected both whole blood and serum. Whole blood was used to analyze white blood cell counts. Serum was used to detect antibodies to Mycoplasma pneumoniae and several respiratory viruses included in the bovine respiratory panel. Mycoplasma pneumoniae bacteria, rather than antibodies, were detected via polymerase chain reaction through nasal swabs, while Pasteurella species were detected by anaerobic culture of tonsil swabs. Upon spontaneous death or euthanasia at the end of the experiment, animals were observed grossly and histologically through necropsy, and we collected lung and lymph node sections to send to Washington's diagnostic lab to detect Mycoplasma pneumoniae by polymerase chain reaction in the tissue. Here we see results for Mycoplasma detection through time post-exposure. The first aspect I would like to point out is that with the exception of one treatment animal, this Aoudad 14 here, as well as the control group, every single other animal tested positive for Mycoplasma at multiple time points. Second, I'd like to point out these indeterminate results shown in yellow here. These can be interpreted in one of 2 ways. Either these indeterminate results manifested because of low levels of detection of Mycoplasma, or they were manifested through low nonspecific replication that occurred during the assay. Because every one of the animals that tested an indeterminate result also gave us a positive result at some point in the study, we chose to interpret these indeterminate results as low or weak positives. Lastly, I want to point out that we detected Mycoplasma by PCR in nasal swabs of at least 2 of our control animals. I'm suspect of the indeterminate result here because, again, we do not have other positive results to concur and give us more information about the shedding dynamics of that animal. This figure depicts the seroconversion and development of antibodies of these Aoudad to Mycoplasma pneumoniae. I would like to first point out that all animals in the Mycoplasma group and the wash group developed antibodies to Mycoplasma, indicating that they received true live bacterial exposures through their inoculations. Secondly, I would like to point out that 2 of our control animals also mounted antibody responses at the end of the experiment. This strongly suggests in combination with our detection of mycoplasma in nasal swabs from these animals, that they received natural exposure to mycoplasma through their fence line contacts with their neighboring treatment groups. Lastly, I'd like to point out that Aoudad 2 from the MOVI-only group and Aoudad 9 from the WASH group both arrived to the study with antibodies to Mycoplasma ovopneumoniae, indicating that they had previously been exposed. I will address the implications of this finding towards the end of the presentation. This figure depicts the Pastorella detection rates compared between the two groups through various sampling points in the study. We can see here that there's a great variation of Pastorella detection both within each group and across time periods within each group. This suggests that Pastorella shedding is more variable than that of Mycoplasma, and suggests that the presence of Pastorella in the inoculum for the WASH group did not significantly increase their shedding of these pathogens. I also would like to point out that there are only two significant differences between the groups in Pastorella rates, and they were reversed in their direction. What I mean by this is that the first significant difference, we can see that the nasal wash group is much higher than the MOVI group, whereas the situation is completely reversed later in the study, where the MOVI group has much higher rate of detection for Pastorella than the nasal wash group. This may reflect a challenge in detecting these species by culture, but it also warrants me reminding y'all that several species of sheep, including bighorn sheep and domestic sheep, typically harbor Pastorella species in their lungs and are completely healthy and fine. So this might play into this intermittent shedding that we're finding here. We use the measurements of total white blood cells, neutrophils, platelets, and fibrinogens to give us an index and an idea as to the severity of the immune response mounted by these animals in each group. We found that there was no difference in the severity of any of these parameters through any point in time between these two groups. The same was true for clinical scores. We found no difference in clinical scores between the groups when compared through time across the study, given the same number of days post-inoculation. Here I have presented the gross and histological responses of each animal throughout the study. The major aspect that I would like to first point out is that we saw 40% of our MOVI-only group, 80% of our WASH group, and 1/3 of our control group display gross and histological signs upon necropsy consistent with pneumonia. These differences were not statistically significant, despite the fact that proportionally they are quite variable, and this is due to the relatively low sampling size. Finally, I want to point out that Mycoplasma ovopneumoniae in tissue was only detected twice in our Mycoplasma-only group and twice in our nasal wash group, while leukotoxin in Pastorella was detected in one of our Mycoplasma-only group animals and 3 of our wash group animals. This slide shows necropsy findings for 2 MOVI animals on the left and 2 wash animals on the right. Our top animal here has evidence of an ongoing pneumonia given by its cranioventral congestion and coupled with its fibrin deposition. This is in contrast to the image on the lower left, which is an Aoudad from the same MOVI group that has pink, healthy appearing lungs coupled with fibrin deposition, indicating it's healed or healing from pneumonia. On the top right in the wash group, we see a lung with heavy fibrin deposition, congestion, and necrosis, indicating an ongoing pneumonia. And if we contrast that with another animal in the same group, we see some relatively healthy lungs coupled with fibrin deposition. So in short, both groups had both ongoing and healing pneumonias. So taken together, the results that I presented suggest that Aoudad are capable carriers and transmitters of both Mycoplasma and Pastorella species, even after seroconversion. Pastorella presence was not predictive due to its high variability, making it difficult to comment on the importance of this in exposed Aoudad. We detected natural transmission of Mycoplasma to 2 of our control animals, suggesting that natural transmission can occur. Our necropsy results suggest that MOBI may largely remain in the upper respiratory tract where it sets up a pneumonic environment. And we saw this because we did not detect Mycoplasma in a high proportion of our lung samples, but we were finding it in a very high proportion of our nasal swabs. I wanted to address the caveat in our results, and that is that 2 of our experimental animals arrived to our study site with titers to Mycoplasma, indicating that they had already been exposed. So the question became whether the results of our experiment were truly due to our artificial inoculation of these animals. We are currently trying to disentangle this through use of sequencing technology to determine which strains came from where to track transmission through these animals. Regardless of the source of Mycoplasma, however, the fact that all of our animals were capable shedders and ended up seroconverting suggests that Aoudad are capable shedders and transmitters of Mycoplasma. Of course, I just wanted to offer transparency as to where I derived the background information and justification for this study. And with that, I'd like to yield my time, offer my contact information, and welcome you reaching out to me. I'd also like to express my sincere gratitude to Texas Bighorn Society, Dallas Safari Club, and Wild Sheep Foundation for funding this work. It would not have been possible without their support. I would also like to extend a thank you to Texas Parks and Wildlife, as they were critical in coordinating the capture and overall structure of this project.